sigma factor. A sigma factor is a protein needed only for initiation of transcription or RNA synthesis. It is a bacterial transcription initiation factor that enables specific binding of RNA polymerase enzyme to gene promoters. RNA polymerase transcribes genes from a specific region of the gene called promoter. The sequences at -10 and -35 sequences in the promoter region are important for binding of RNA polymerase enzyme for transcription in prokaryotes. The RNA polymerase holoenzyme is found by the association of sigma factor with core polymerase, which helps in the promoter selection at the initiation of transcription. The different subdomains of the DNA binding domain of the sigma factor contact the different regions of the conserved promoter. Different sigma factors are used under different environmental conditions. These specialized or different sigma factors bind the genes in the promoter region in appropriate environmental conditions and increase the transcription of the genes. Prokaryotic core transcription machinery recognizes the promoter sequence, which has -10 and -35 sequences. The extended conformation of the sigma factor binds to -10 and -35 sequences of the basal promoter. The NTD, that is N-terminal domain of each of the two alpha subunits binds to the core polymerase enzyme, while the CTD, C-terminal domain, interacts with the upstream promoter elements. To explain the role of sigma factor in prokaryotes in different environmental conditions, we can see the most extensively studied prokaryote E. coli as an example. Sigma factors in E. coli: sigma 70, otherwise sigma A or RPOD. The function is housekeeping sigma factor or primary sigma factor. It transcribes most genes in growing cells and makes the proteins necessary to keep the cell alive. Second one, sigma 19, the ferric citrate sigma factor. It regulates the ferric citrate gene for iron transport in prokaryotic cell. Sigma 24, RPOE, the extra cytoplasmic or extreme heat stress sigma factor. It regulates the heat stress when the bacteria are exposed to extreme environmental conditions such as heat. Sigma 28 or RPOF, the flagellar sigma factor that directs RNA polymerase to flagellar late promoters. Sigma 32 or RPOH, the heat shock sigma factor turned on when the bacteria are exposed to extreme environmental conditions such as heat. Due to their higher expression, the sigma factor will bind with the polymerase core enzyme. Other heat shock proteins are expressed which enable the cell to survive higher temperatures. Chaperones, proteases and DNA repair enzymes are some of the enzymes that are expressed upon activation of sigma 32. Sigma 38 or RPOS, the starvation or stationary phase sigma factor. Sigma 54 or RPON, the nitrogen limitation sigma factor. There are also anti-sigma factors that inhibit the function of sigma factors and anti-sigma factors that restore sigma factor function.